Okay, so yeah, I think that people now getting in, which is good, on both Zoom and YouTube. So Veronica, shall we start?
Mm-hmm.
Or shall we wait for a few more minutes? I think we have a good number of people now. Okay, brilliant. So yeah, sorry for the technical challenges that we're having now, but I think we have solved most of it. So I'm very happy to start our first seminar this year, 2021, on the CDT, Agriforwards CDT Science Seminar Series. And I'm very excited to have Professor Alison Smith. She's a professor at the Department of Plant Science, University of Cambridge. And I have known her quite some years because we belong to the same college, but we never really talk about this main business for a long time. So I'm very much looking forward to what's up to in her work in the area of plant science, especially the plant biochemistry. So without further ado, I'd like to pass the floor to Alison, and then we can have some discussion after the lecture. So I stop sharing my screen and hand it over to Alison.
Great. Thank you very much, Famia. I'll just share my screen now. So I hope you can see that.
Yes, I think you're good to go. Maybe you can make it the full screen. Yeah, I think this is perfect. Thank you very much. Looking forward to your talk.
Great. Okay, so thank you very much. And I hope that this will be of interest to you. As I've mentioned already to Famia and Veronica, not a single robot is involved in this, but there are a lot of organisms. So what I'm going to do is to tell you about something about algae, explain what they are, and tell you how we think they have potential as a food ingredient, how we can enhance the nutritional value of food using them, and how we might be able to reduce waste in the process. So firstly, I will, once I've explained what algae are very briefly, I'll tell you what the opportunities are for the exploitation of algae. Then I'll explain features of algae as food. I'll tell you about the importance of vitamins, which is my research area. And then I'll tell you about some of the things that we've been trying to do to think about how we might incorporate cultivation of algae to reduce waste and indeed valorize it. So firstly, I should explain what algae are. So these organisms are eukaryotes. So that means cells that contain a nucleus. And they're mostly photosynthetic, although there are some non-photosynthetic ones. And they're found in all freshwater and marine environments. In fact, they're found in all environments around the globe. In one of my projects, we study them living on snow in Antarctica. They're also, you probably come across them as a sort of green slimy stuff on your patio, which appears just after rain. Now, there are two very broad categories of microalgae. I should just make my, sorry, get my laser pointer. So on the left down here, you can see these are macroalgae or seaweeds. And so you might be familiar with them from the seashore, like bladderwrack here, or lava. And lava is also used in, for making lava bread. On the right, I've shown a number of different examples of microalgae, and it's indeed microalgae that we study, and that's what I'll be talking about for the rest of the talk. I should say, so I won't go through them. They've all, I think they're very beautiful. They've interesting shapes, interesting behavior, nice colors, but they've got very complicated names, so I'm not going to bamboozle you with them. One thing, though, I should say is that they are incredibly diverse, so they're very, very, very variable. And so even though to you, or to engineers, they might look very similar, small cells, this organism here is a diatom, and this is a green alga. And these are more dissimilar to one another than we are to fruit flies or indeed fungi. So you have to think of them as not just one category. One thing I should say, and this is of relevance, I'll show you a bit later on, is that cyanobacteria are not algae, they're bacteria, so not eukaryotes, they don't have a nucleus, but they are functionally treated as such, and indeed in legislation are very often lumped together with algae. So why are we interested in them? So this is about the cultivation or the commercial exploitation of microalgae. There is interest in commercial exploitation of seaweeds, but I'm not going to talk about that, as I've said. So firstly, they're photosynthetic, but they're a non-food biomass. So they don't compete for agricultural resources. They don't compete for arable land. They don't compete for water, fresh water resources. Interestingly, it takes less water to cultivate these aquatic organisms than it does to grow land plants, because land plants need a supply of water continuously. Many microalgae have very fast growth rates, so their productivity can be 20 times that of land plants. And that's because the material that you might want to harvest is the entire cell as opposed to one part of the plant. Oops, sorry, something happened then. In addition, they make many of them make very interesting and valuable compounds, and indeed the exploitation that is done for these organisms is for making things like pigments, vitamins and antioxidants. And the advantage of microalgae over land plants is that they can be cultivated at scale in fermenters, and I'll come back to that a bit later on in the talk. So this is something straight. Oh, so this is my student producing clever animation here, not telling me. So these are images of cells that are accumulating lipid droplets. So there's a single cell, and it's a fluorescence image. You can see red, that's the chloroplast in the cell, and then these yellow spots around the outside are lipid droplets. And over time, grown in low nitrogen conditions, the photosynthetic products are accumulated in lipid droplets. And this phenomenon is well known and had been studied a great deal in the 20th century. In the beginning of the 21st century, it was proposed that you could use this phenomenon of accumulation of massive amounts of lipid. It can be up to 70% dry weight in weight per volume in a unicellular alga. Could be used for biofuels, because this is neutral lipid and it can be transesterified to produce biodiesel. And so this is an estimate of the potential oil yields in litres per hectare. And so these are actual values for palm oil, rape, soybean, and jatropha, which is a tropical oil seed plant. And then on the right for Nanochloropsis, which is a unicellular alga. Now this green number here, this is the theoretical potential yield, because I don't think anybody's been able to grow Nanochloropsis at a hectare scale, let alone the thousands of hectares that would be needed for making biodiesel. Nonetheless, you can see it's an incredibly, potentially incredibly productive organism in terms of this yield of oil. And a long time ago, together with Stuart Scott from the engineering department, we did the back of the envelope calculation to work out how much microalgae you'd need to grow to produce enough biodiesel for the UK. So this is the current, current, that was 10 years ago. It's probably gone up and then gone down completely because of the pandemic. But anyway, that was the UK diesel use in 2010. This would require 17.5 million hectares of oilseed rape, or 0.8 million hectares of this microalga, if you could get that yield. And this is the UK land area. So there are some people who would say that it would be impossible to make biodiesel because there's not enough land on the planet. That's not entirely true. It's just that we use the land for other things like growing food and other forms of production as well as living. Nonetheless, this is it. You can see these figures imply that algae have a much higher productivity. So this got everybody terribly excited in the noughties, but unfortunately, basically algal biodiesel is not commercially available now and probably won't be in the near future. And that is because the biofuel pipeline, that is to grow the organisms at the scale that you need to generate enough for biodiesel, to harvest it and to process it in order to make the biodiesel, is complicated, and it requires a huge amount of energy input, and it's marginal whether you'd actually get more energy out than you'd put in with current technology. But more, so you could optimize it. Perhaps there's an opportunity for researchers as in your CDT to think about this, but the price of oil goes up and down, and it's very low at the moment. So economically, it's just not viable to put a lot of effort into growing algae at scale. And indeed, it always seemed to me that being wanting to grow thousands of hectares of algae when we didn't really know how to grow them much outside the laboratory was perhaps a little ambitious. And maybe we should think about growing them for something that you would grow at a lower volume, but maybe a higher value. So in my lab, we're actually interested in growing microalgae for production platforms for high value chemicals. I'm not going to talk about that at all today. Instead, I'll talk about something which is slightly lower value, but nonetheless, you don't need to produce at such a scale as you would for biodiesel. And that is for food and feed. And there are lots of features of microalgae that are good for as a feedstock for producing food. Firstly, because they're photosynthetic, they have a very high protein content. The proteins involved in photosynthesis with a balanced amino acid profile, or sorry, it should say amino acid profile. Many seed proteins, which are being actively sought as an alternative to, for example, dairy protein in the food industry, have a slightly biased amino acid profile because they're storage proteins. But that's not the case for microalgae. So they effectively have the same amino acid profile as leafy food products. They also are a source, or many species are a source of essential fatty acids such as EPA and DHA. These are omega-3 fatty acids. Indeed, fish oil contains these compounds because fish eat the microalgae that make them. Many algae are classified as grass, which is generally regarded as safe, and therefore are potentially could be legislated for or licensed for food. And they also have a very high vitamin and mineral content. And this is really important because in thinking about the Sustainable Development Goals, whilst hunger is really important, it's not just calories that are necessary to sustain life. You also need sufficient nutrients in your food. And in recent decades, the focus has been on producing or increasing yield to the detriment in certain cases of quality in terms of composition of protein, of lipid and micronutrients. And so thinking about things like good health and well-being, which requires a balanced diet, is very important in trying to achieve these goals. So I should also just take an aside to explain what vitamins are. This is, as I mentioned, this is what I'm interested in my research. Vitamins are organic micronutrients which are required for growth. So these are compounds which we need to take in our diet. But actually all organisms need micronutrients. It's just some organisms make them. So these oranges are making large amounts of vitamin C, but they're not making vitamin C for us. They're making vitamin C as an antioxidant and as a cofactor for enzymes in the plant tissues. That's important to bear in mind. So vitamin was a term that was originally coined as vital amine, and then it was concatenated for thiamine, which was the first vitamin that was identified, and it was isolated from the husks of brown rice, and it cures beriberi. So beriberi is a deficiency disease when you have insufficient thiamine in your diet. There are lots of other vitamins though, and there are characteristic deficiency diseases for all of them. So vitamin A deficiency, this is vitamin A, I think. I can't see over the screen of my pictures. So I think this is vitamin A here with a hydroxyl group on the end of it. So this is an important compound for the retinal pigment in our retinas. Folate, which is this molecule down here, deficiency in that leads, for example, to neural tube defects. And then this vitamin here, vitamin B12, is the most complicated vitamin. It's actually the most complicated primary metabolite known in nature. And its deficiency is linked to ischemic heart disease and cognitive impairment. The disease is, or one form of the disease is called pernicious anemia. And it's this vitamin which I'm going to tell you about in the context of using algae to provide it. So as I've mentioned, vitamin B12 is also called cobalamin, and that's because it has a cobalt atom in the middle of the molecule here. The deficiencies, as I've said, lead to neuropathy and malaise associated with cognitive impairment and decline. It's not made by plants. It's not made by land plants. It's only made by bacteria. So very strict vegetarians and vegans are at risk of deficiency, serious risk of deficiency, and are strongly recommended to take B12 supplements or injections. In addition, in the elderly, there's a reduced ability to absorb the vitamin, and I'm shocked to have to tell you that the elderly means over 50. And so again, there's also you're recommended to think about your source of B12, particularly if you don't eat a lot of red meat and dairy products. And indeed, there is some link between cognitive impairment and B12 deficiency. So if you find yourself forgetting where you put your keys, think about taking some B12. So what's that got to do with algae? Well, it actually turns out that many algae which we already eat have very high contents of B12 in them. So this is data for porphyra and pyropia. So porphyra is larva, and that's used for larva bread, for example, and pyropia is nori, the source of seaweed which is used in sushi. And then this is a summary table of vitamin content for porphyra versus liver. And these are vitamins A, E, and C. I won't go through those now, but you can see for vitamin B12, there is about 70 micrograms of B12 per 100 grams dry weight of porphyra and 110 in liver. So liver is the recommended source of B12 if you are deficient. So if you don't like eating liver, you could eat seaweed instead. But in fact, microalgae are also a good source of B12. So there are actually 2 microalgae, in inverted commas, which are licensed in the EU as novel food, and that is chlorella, which is a microalga, and then this cyanobacterium. Remember I said cyanobacteria are not really algae biologically, but they're functionally classed as such. And this is sold as spirulina. So you might be familiar with that. It's actually got this beautiful spiral filamentous structure. So both of these organisms have high protein content, high mineral content, and many vitamins, including B12. But the interesting thing here is that spirulina can synthesize B12 because it's a bacterium. But chlorella cannot synthesize B12. So there's no known eukaryote that makes B12. It's only the only source of biosynthesis is known to be from bacteria species. So this was quite surprising to us when we first started to measure it in foodstuffs and found this. But one thing that we've been looking at in my laboratory is the fact that there are many algae which actually are like animals. They need a source of B12 to grow. And we showed that they can do that by growing in symbiosis, in mutualistic interactions with bacteria. So here's an experiment that we did some time ago with an alga called Lobomonas rostrata. As I told you, they've all got funny names. And it's growing in a flask with B12, and you can see it's nice and green. If we grow it in the same medium, but with no B12, then it doesn't grow at all. But if we include a bacterium, Mesorhizobium lotti, a soil bacterium in the culture, then that supports the growth of the alga, so it's green. The bacteria can't grow in this media either. So that's because, so they can grow here and provide B12 to the algae because the algae provide photosynthetic. They provide, we don't actually know what the molecule is, but sugars or amino acids in return for the vitamin B12. And these interactions are actually typical of microbial communities. And this is an experiment where we took different proportions of algae and bacterial cells. We initiated them. So for example, we initiated a culture with 10,000 algal cells per bacterial cell, or 100,000 bacterial cells per alga. And then we grew them over several days, and they reached this stable equilibrium of about 30 bacteria per algal cell. And this is characteristic of microbial communities. This is a very simple community of 2 species, that there is a mutualistic exchange of compounds between the organisms, so they regulate one another's growth. So what we wanted to do was to see whether we could use this approach to try and increase vitamin B12 bioavailability. And Chlorella vulgaris, which is this this licensed microalga I mentioned before, my student Ellen Harrison was able to show that it can take up vitamin B12 or cobalamin from the medium. So this is just a simple experiment having increasing amounts of cell density, and we incubate them with a fixed amount of vitamin B12 and then measure what is present either in the media in glorious purple or in the cells in green. And you can see when there are very low levels of algal cells, then there is a little bit of taken up by the cells, but as you increase the number of cells, and then you can see that there's a gradual increase up to a sort of saturation here of B12 being taken up from the medium. So that is when we've added a chemical form of the vitamin. but what if we grow it with bacteria? Well, we've now been able to grow it with grass bacteria and show that the cells take up B12 from the medium in the normal way. I can't show you the data for that at the moment because we're still in the process of publishing it with our collaborators. Nonetheless, we have started a project with colleagues in India, in Pune, looking to see whether we can take this approach to increase the availability of B12 to the local population. And one of the characteristics about the Indian diet, and in fact the Indian population, of course, is a very high proportion of vegetarian and vegan people in India. In addition, many people who are living with very low income streams can't actually afford to eat much meat or milk products. So the idea here is to see whether we can generate algal biomass by growing it with edible bacteria, and then test to see whether the algal biomass that we've produced can actually make a difference to the B12 levels in a population. So we were awarded a GCRF grant, again, along with my colleague Martin Warren in the University of Kent. We prepared, well, we didn't prepare them, but we provided the algae to prepare delicious looking green food bars. And then these were used in a dietary intervention study in Pune. And again, I can't actually show you the data directly, but I can tell you that there was an increase in B12 levels in the serum of the individuals that were in this study, which was very encouraging. So this was increasing or providing them with a green bar once a day, 5 days a week, for 6 weeks. In March 2020, we were all set up to have a much larger dietary intervention project of 600 people to take place over 4 months. Unfortunately, something happened in March 2020. Basically, we weren't allowed to do anything for a while, and then people in India are not allowed to do anything for a while. We are now having a conundrum of not really knowing whether we can go ahead ethically with this trial, because B12 is now being prescribed along with many other vitamins as a way to reduce the likelihood of infection with coronavirus. And so we will not be able to have a population where we know what their B12 status is before we start the project. So that's slightly unfortunate, but we do think that there is an opportunity to consider providing algal biomass for foodstuff as for B12 production using this approach of growing them with edible bacteria. Undaunted, and actually as part of the project, we have also been working with the Melissa Foundation, which is funded by the European Space Agency, and we're taking this approach to try and see whether we can provide vitamin B12 for astronauts on long-haul space missions. So the Melissa project is actually trying to work out how to have a completely closed circle of microbes to provide all of the nutrients and deal with all the waste streams for astronauts on these missions. And there are a number of algae and bacteria which we have started to use in different combinations. So that's all I wanted to say about food per se, but I wanted to just give you a little bit of a sort of where else you might be able to incorporate the use of microalgae in the agricultural process. And so I should start by showing you this facility, which we have in the Botanic Garden. So this is an absolutely massive or huge greenhouse, which is in the Botanic Garden in one corner of it. So if you know the Botanic Garden in Cambridge, you would come in here from Hills Road, and then you'd see this very big greenhouse. And this is our Algal Innovation Centre. And basically it enables us to test a whole variety of different approaches to develop pipelines for algal-based solutions. And this enables us to grow algae under natural conditions, essentially natural conditions, because it's open to the light. But it's contained, so we're able to grow non-native algal species and also genetically modified species, so that's not relevant for food use, but rather for high-value products. So this facility enables us to interact with a number of different companies who are interested in a whole range of different, things, different ways of growing algae, harvesting algae, and so on. And as a result of that, we came up with some other ideas about being able to use our microalgae in small modules, which can be combined in different permutations. So here is how we actually grow the microalgae. This is my colleague Matt Davey, and he's pouring a small flask of algae here into a larger, this is a tubular photobioreactor of about 10 litres, and you can see it growing here. We have much larger photobioreactors, which are about 150 litres. These circulate, turn around and circulate and allow the biomass to produce. In collaboration with a small company from Wales, Membranology, we've now got this membrane concentrating system here, which enables us to harvest the biomass without having to centrifuge them. And then we end up with pellets like this. And if you are familiar with what algae look like, which you probably aren't, you can see that there's a dark green lump here, and then there's a slightly lighter browny coloured material there, and that's the bacteria which are growing with our algae. And that's because this is open, we don't try and keep it sterile, and algae and bacteria have a natural propensity to live together. So what can we do with that algal biomass? Well, we have been working with a number of people in developing countries, and in this particular project with people in the University of Ghana in Legon, Accra, they came to us and said, we have this issue with crop waste. And crop waste, I don't mean waste in the, oh, sorry, somebody's trying to call me. I don't know how to stop that. They have I don't mean crop waste in the field, I mean once it gets to market. And that's because this is the way in which crops are transported in the back of small vehicles, and usually the crops at the bottom, or the harvested fruit at the bottom of the vehicle, is the packing for the rest of it. So when this is offloaded at the market, all of the bruised and damaged fruit are basically thrown away. So the markets in the city are full of rotting fruit and vegetables, which are unpleasant, attract vermin and so on. So they're interested in trying to work out what to do with that. And so we're working with them to try and implement anaerobic digesters in the market, which will provide biogas or methane. But then you're left with a very nitrogen and phosphorus rich digestate. What can you do with that? Well, you can grow microalgae on it. Microalgae require these nutrients to grow. And as a result of doing that, you end up with algal biomass and cleaner water. I mean, it's not potable water for human consumption, but it is a lot cleaner than it would have been there. And then this water can be um, applied to the fields or put into the watercourses without worrying too much about eutrophication, too much nitrogen and phosphorus. But we've taken this a step further, and together with colleagues at NIAB in Cambridge, which is the National Institute of Agricultural Botany, and a small SME, Agrigub, we are using the algal biomass and feeding it to insect larvae, which then provides animal feed. And these insect larvae are shown here. So these are black soldier fly larvae, and Agrigub are experienced in growing them. And normally these these larvae are fed on orange waste, and a few other nutrients. And we've been able to show that we can substitute the other nutrients for with algae, and the grubs grow very well and as effectively as with the defined medium that were being used before. And we're now in the process of trying to determine whether the vitamin content, which we think this algal biomass contains, is passed through into the grubs and then ultimately to animal feed. So this is a sort of summary of where we, you know, the sort of things that we're thinking about that we might be able to do here. So as I showed you, we have crop waste, which can go for anaerobic digestion, to make digestate, allow algae to grow, and then we can support the insect larvae. And we have products that come out in all of these positions. But in addition, insects produce waste, and that's known as frass. And frass is very high level of chitin, which is a polysaccharide molecule, which has substance. And this is used already as a soil improver, so that increases the structure of the soil. And then that facilitates crop production, which then eventually would lead to crop waste. And so you'd sort of close the circle. And the bacteria that we use for supporting our algal growth and providing B12 are actually soil bacteria. And these are important for improving the nitrogen fixing and phosphorus uptake within the crops. So we can close the circle like this. We can actually even consider growing microalgae using different waste streams and then use them in anaerobic digestion, and we've shown that that works as well. Or we could use them directly as a fertilizer onto the soil. And so together with NIAB and other collaborators, industrial collaborators, we're trying to work out how to optimize each one of these modules, so this part here, this part here, and so on, and then enable them to be combined, mixed and matched in the appropriate way, depending on the inputs and outputs. So your inputs might be brackish water, might be nutrient enriched, it might be low-grade heat, and your outputs might be food or feed, fertilizer or soil improver, or cleaner water, for example. Okay, I think I've talked for long enough, so I should just summarize by saying that algae, particularly microalgae, are amazingly diverse, and with a bit of imagination, they offer the potential for many potential avenues for commercial exploitation, possibly for biofuels in the future, should the oil price increase. What I've shown you is that vitamins are important for nutritional value, and vitamin B12 in particular is unique because it's not made by plants, so you can't get it in a plant-based diet. my interest is trying to understand the microbial communities, and it's actually much more than just ecology. It is as much to do with serendipity and thinking about where you might employ these microbial communities. And in terms of trying to do something which you can claim is sustainable, it's probably a good idea to think about considerations of sustainability from the outset. So rather than coming up with a process, optimizing it, and then worrying how to make it sustainable, I think with the potential that I've shown you for different modules using microalgae, there's a possibility for having a sustainable process before you start or when you start, rather than having to retrofit it. And I should just end by saying thank you very much to my group. This was Christmas 2019, when we were all allowed to associate with one another. But in particular, I'd like to thank Matt Davey, who was instrumental in setting up the AIC, and he was the person who was growing the algal biomass. Payam Shahi, who's now the lead on this and the manager of the Algal Innovation Centre, and Ellen Harrison, who is working on the Melissa project for algal bacterial combination for B12 delivery in space. And thank you very much for listening. I'm happy to take any questions.
All right, thank you very much, Alison. This is an amazing talk. I really learned a lot of things in this very short time. And I knew nothing about this in detail, but I think this was wonderful. I can't help asking, how does it taste to eat algae?
It doesn't taste of anything really. So it depends. So some, if you treat it in a particular way, particularly if you heat it when you're harvesting the algae, you can get a slightly sort of grassy flavor. But certainly if you take chlorella tablets, then basically there's very little taste in them. Spirulina does have a slightly well, I don't know what to say. I think it tastes slightly musty, but you can buy all sorts of things that contain both chlorella or spirulina. You can buy smoothies. You know, there's innocent smoothies and other brands available that are sort of blue-green. That's the cyanobacteria content. But they have lots of other things in them, so you can't really taste. You can't really.
Okay, thank you. Let me just ask one more question from engineering point of view. We're very much interested in if there's any engineering challenges in the production of this algae. Do you have anything in your mind that you could suggest for potential engineering challenges that we could contribute to this project?
Yeah, I mean, I didn't, so we buy our photobioreactors and we collaborate with companies who make them. So one of the major things is that they need proper light penetration. So if you have, in order to get the maximum yield of microalgae, they can't be too dense. So you have to have light going through into the middle of the culture. And they are so efficient at absorbing light that cultures that are more than, say, 1 to 2 centimeters deep will attenuate all of the light. Or, you know, so if you have a culture that's 10 centimeters deep at the bottom, it will not have any light in it. It will have sucked it all up. So you have to circulate it. And that's where the energy input goes in. So even though we don't need the same volumes for these food products, we still need to put quite a lot of energy into cultivating them. So thinking about engineering solutions for that is definitely one. The other aspect is harvesting. So these organisms are very often about, let's say, 10 microns in diameter, round, very slippery, and quite difficult to, you know, pick up. They are, and then actually breaking them is difficult. So yes, there's a lot, there's lots of opportunities. I just don't know if there's any opportunities for robots.
No, that's brilliant. I think there are lots of interesting questions that we could potentially discuss. And there's one question in the Q&A window that what is the energy and materials footprint for algae compared to other food stocks?
If you just need to say, sorry, I've promoted everyone as a panelist, so people are most welcome to ask questions themselves.
Okay.
Thank you.
So shall I answer that question in the chat? I can see the chat. It's much worse than for land plants. Okay, and that is because we've had 10,000 years of working out how to grow land plants or crop plants. So we're pretty good at it. I think if you said, what was the energy input for growing wheat until, you know, coal was available, I think it would be quite a lot. So it's not optimized. The largest algal facilities are about tens of hectares, and they usually are for microalgae that have they're extremophiles, so they can tolerate high salt or high light or high pH, which then prevents the adventitious organisms, which, as I've tried to point out, microalgae like to live with other organisms, but that then impacts on the yield of your crop. So it's not possible to directly compare the energy and other inputs into growing microalgae with growing crops, because we just it hasn't been optimized. That level of agronomy is not known. I know that's a bit of a cop-out, but it is. I think the corollary for that you can say is that you can grow them on waste streams in ways that you can't for those so much or not so easily as for crop plants. So you can incorporate or enhance or reduce the cost, shall we say, of the inputs by using waste streams which you would otherwise have to get rid of or don't have to pay for.
Simone, do you have any follow-up question? Are you happy with that?
No, that sounds really interesting. It sounds like an opportunity maybe to look into that and see what could be done. And yeah, obviously it's a huge industry and a growing industry, so there might be something that can be done.
Okay, thank you. Fulvio, do you have a next question?
So are really those the limiting factor? Because from what you presented today, it seems that there is a huge potential, and one wonder why it's not already out there. I mean, it's in terms of protein production, chemicals, density of production. So what are the limiting factor of the explosion of the field, essentially?
Okay, I think there are two things. One is, I think, Fumiya asked me, what does it taste like? It actually looks green. There is a small company that has recently been able to generate an algal strains that aren't green. And so you could contemplate incorporating that protein, organism as a protein into bread or other products and not have them go green. I mean, there are other ways to get rid of the chlorophyll, but the flavor is also an issue. It's novel. And so in terms of food, there's a sort of social and consumer angle that has to be taken into account. So it's actually trying to persuade a manufacturer to agree with that. That's one side. The other side, so basically, those of us who are involved in this is we are aiming for hero products. I think that's a well-known term. Well, I'd never heard of it before, but a hero product, in other words, something that you can use to show the value and the advantage of a particular approach. But it's just, what might that hero product be? Well, I think as you might have gathered, vitamin B12 as a source of vitamin B12 is a unique one. But the other thing is regulation. And it is actually very, very difficult to introduce novel foods. So one of the things that you can do with any crop plant is you could think of a novel thing to do with that crop plant. But that's because it's already been licensed in the EU, and also it will be subsequently licensed by DEFRA or the FDA in America. Whereas for microalgae, you can only use, or for algae rather, you can only use things as novel ingredients or feedstocks without applying for licensing if there's a historical precedent. So you can do that with seaweed, because with various types of seaweed, because they've been used for centuries, and so it's then acceptable. But for any sort of novel, sorry, for any new microalgae, it's regarded as a novel food, and it would have to be licensed. And it's, you know, it's prohibitively expensive. None of the companies that are involved in doing this are big, and therefore, you know,
if I can continue, I was a bit curious about this futuristic application in aerospace. Besides food, what about production of oxygen? Given the density, it's something that is plausible or feasible, or it's something that is no?
Well, in fact, the Melissa loop, it's called, is uses land plants to produce the oxygen, and also I think they have the whatever it is, I don't know how to split water. I don't know how they do that. Electrolysis, I guess. Yeah. So it's they couldn't rely on plants to produce all of the oxygen.
But algae seems to be a higher density, so yes, that's why.
Yeah. So the Melissa loop was conceived, I think, about 30 years ago, quite a long time ago. And they have these various modules, which are, or compartments, each of which is responsible for one part of the life support system. And the oxygen it's like a supplement. The plants can produce oxygen as a supplement, but it wouldn't be the only way of doing that, because it's too risky, I suppose. And so microalgae were not involved in the loop until we were involved with it. So we've now tried to introduce that. I'm still not convinced that you'd be able to produce enough oxygen to sustain all of the activities without supplementing it with other things.
But you think it's definitely more than plants, way more than plants?
Yeah, well,
yeah,
the productivity, and as engineers, you'll know what I mean by productivity. Biologists don't, but you know, yield per unit area per unit time is higher than for crop plants, but you do need it. It hasn't been demonstrated to be able to be maintained continuously over a long period. So I think I think the longest cultivation period is about, well, on average is 3 to 4 months that people do before they have to restart it in commercial production, and that's not robust enough as an oxygen supply.
Brilliant. Yeah, thank you very much. There's one last question. Can algae be used in the context of harvesting energy like solar panels? And I think this is probably in the context of a biofuel route, if I understand correctly. Would you answer in the same way, Alison?
Yeah, so I think there is a lot of interest in using the photosynthetic capacity of microalgae in photovoltaic panels, for example. There are projects in Cambridge looking at that, and also to harvest light energy on buildings, actually then to cool them. There was a house, Algae house it was called, in Hamburg, I think, in Germany, that was set up with that in mind. So they had panels on the front of it where algae were grown, that reduced the light that was falling on the building, so you needed less cooling. And then the idea was the biomass was then subsequently used. Unfortunately, it didn't last for very long because I'm fairly certain at least part of it was contaminated with bacteria. So it's a bit of a you know, common theme here. But there is a lot of interest in actually incorporating algal growth into architectural or building design. If you think about cities, and we have talked with lots of architects and engineers about this, cities have got lots of flat surfaces which are not used. And you can put solar panels on them. But actually solar panels can, there's interest in having a solar panel, then underneath it growing algae on the light that is not absorbed by the material in the photovoltaic. And to be honest, I don't remember what that is. It blue? I think it might be blue light. So as long as there's some red light going through to the algae, they would be able to grow. So it's things like that. I think there's a lot of potential, but all of the energy uses. Remember, you don't want something that requires more energy than you get out, because that would be pointless. But also, you do really need to do it at a much bigger scale than you do for the things such as food or other chemicals.
Yeah, thank you very much. I think this is really, really exciting to hear all these futuristic technologies, and I'm pretty sure lots of students in CDT looking for problems, engineering problems in particular. So I'm sure Alison is available for further discussions for this kind of challenges if they have questions. I think, yeah, that's all for today's seminar. Well, thank you very much once again, Alison, for a wonderful talk today. And we learned a lot of things, and hopefully we can develop very good collaborations within the CDT context. And we will have a next seminar on the February 19th. So we will see you once again in a month's time. And thank you very much, everyone joining this webinar today. Thank you very much.
Pleasure. Bye-bye.